Today I have the pleasure to introduce you to Professor Biancampa, who is a professor here in the RWTH University now and in the biology department. And yeah, he will talk to us a little bit more about doing the hands-on work in the experimental side and the neurobiology side. Bjorn, we are very excited to have here this nice group of young, motivated scientists. They come from all over Europe today. They are between the master, some of the masters, some of the PhDs, some of the postdocs. They are all very excited to learn more about neuroscience and how to apply EduBrains to also address questions in the direction of neuroscience and brain medicine, with a very diverse background as well. So,
nice.
We thank you very much for your availability to give this talk.
Yes.
And I guess, yeah, I think we are almost on time. So
okay, I can start. Yes.
Thank you so much.
Thank you, Sandra, also for the invitation to speak in front of this crowd of very diverse students and postdocs apparently. So unfortunately, I don't have the time to talk to each one of you, but if you have questions during my talk, please feel free to interrupt me. And I will try to do something like a semi-lecture versus science talk. I thought that might be most appropriate for this audience. And there's also the link to our website. If you have questions or you were too shy to ask now, or you come up with questions or ideas later, feel free to contact me. Then we can interact also afterwards. So when I was sitting, maybe like you as a student, starting out thinking about what I want to do in science, I turned out that neuroscience was what I was really interested in, and mainly because I wanted to know how the brain works, or more specifically, the cortex. And as a biologist, I was mainly interested in how individual neurons work. So this is the cellular mechanisms that are in the title here. So neurophysiology basically means that you want to understand the function of the brain and not just how it looks like. And so this is how this maybe somewhat strange title comes about. But I'm, of course, not the first one who is interested in that. Turns out that humans have been interested in the brain already for a very long time. And this skull here that was found in Alsace in France. Anyone from France here? So from your home place, maybe, in Alsace, which is actually at the German border. And so this was a French person, obviously, about 7,000 years ago. And you can clearly see that there is a hole in the skull. And it turns out that this has healed. So the patient was actually alive when this hole was done, presumably awake because there was no anesthesia yet, and also survived that for quite some time. So obviously, already back then, humans had an idea that the place of our spirit and mind might be inside our head. And presumably this patient had maybe mental problems, and they tried to relieve him, or I think it was a man, from these problems. Now, drilling holes in skulls is actually what we still do today in order to get to the brain. And one example from or one pioneer in this technique in the last century was a guy called Walter Panfield, a Canadian neurosurgeon who, similar to what we do today, had to open the skull in order to cut out pieces of the brain, for example, a tumor. And or if you have untreatable epilepsy, then the focus of this epilepsy will be cut out. This is actually what also happens here in Aachen. As a matter of fact, we had a surgery yesterday. As I they stimulated the surface of the cortex just with electrodes pushing on it, and the patients were awake, and so they could ask them what they feel, if they see something, feel something, experience something. And this you see in this video now.
In response to this stimulus, which they were quite unaware of, and they were in the operating room, you were given a diagram.
I have a funny feeling, as though I went through all this before.
Now I see them. They're laughing. And I'm looking at how they've done better. They're all fine.
I have a children, Marcy. I think we've done all the time.
And so on and so forth. So when I was a student, there was no YouTube yet. So this is a YouTube video, but I was shown this video in one of the lectures, and I was amazed that you can stimulate a very specific memory that comes up from years ago when you were in South Africa and whatever, just by pushing on the cortex in some very specific point. I mean, this might spike some movies that you might have seen recently, but the idea is not that far-fetched, that these memories sit somewhere in this location in the activity of individual neurons. And Penfield has realized the importance of this work and has actually systematically mapped all these brains that he got and all these locations that he recorded from, as you see here in one of his original drawings. And this is also the basis for something that we call nowadays the homunculus. And this is for the sensory cortex, somatosensory cortex, where you see individual body parts mapped onto the cortex where the neurons are that would respond to that. In the same way, you also have this for the motor cortex. So this is the primary sensory area for our body and the motor area, but you also have other areas, auditory and visual areas. This is our speech area. So this is something that we do not want to destroy if we do such a surgery, hence the reason for this stimulation that Penfield did. Now, especially the visual cortex turned out to be an area that was heavily investigated, mainly in animals, so at that time cats and dogs, and for the simple reason that it's easy to stimulate it to just show pictures or visual stimuli. And this is what you also see in an example from a recording in an anesthetized cat. And so the sound that you hear, this crackling sound, these are actually individual neurons recorded with an electrode that is sticking in the brain. And in this case, in the visual cortex of an anesthetized cat. And as you saw in the stimulation, these neurons respond to this light bar of a very certain orientation and a very certain location. And this is what these two guys found out in mid last century, Hummel and Wiesel, and they mapped out the visual cortex of this cat very precisely, and not only found that they are orientation-tuned neurons, but also that they are arranged in a certain organization. It's not just randomly scattered, but as you see here, that neurons with the same orientation tuning are usually placed above each other. And as you move along the cortex, they slightly change their orientation tuning. So this was then coined a columnar organization because of all the columns. And even these columns are arranged in a certain order. So this whole thing gives you a so-called hypercolumn. And this was ground-laying important work, obviously. That's the reason why they actually also received the Nobel Prize for this work. And this is now basic knowledge that you find in the textbooks, like the latest edition of Kandel, where I took this picture from. they mainly worked in the primary visual cortex and also found out how inputs from the thalamus and the retina actually end up here, but also they could show how higher visual areas are then how these are arranged and how they receive the information through the visual cortex. And here are two of these areas, MT and V4, but there are many more of these, as shown in this very nice scheme from Velleman and Van Essen. So this is one of the icons, if you want, in visual neuroscience. So you have down here the primary visual cortex. So this is the thalamus. You have V4 and MT, these two areas, but you can see that there are many more areas all the way up to the hippocampus, where this information then forms new memories. Turns out that the mouse has something similar, although much simpler, of course. Maybe not surprisingly, because the mouse usually lives underground in darkness, but it does have a functional visual system that is at least similar to ours, which is why the mouse has actually become one of the prime subjects that are studied these days in neuroscience. However, if you stick to the humans for a moment, it is also possible to record from the human brain and also stick electrodes inside the brain. Again, this is mainly done in epilepsy patients to find out where the focus of your epileptic seizure is. You are implanted with electrodes that stay in there for a while, and that allows the researcher or the doctors to also perform experiments with you. And what has been done about 20 years ago in this case, so this is why the quality of the image is not that great. It's a few years back. Nevertheless, what they showed to these patients were pictures of celebrities, because everyone knows these celebrities. Back then, Jennifer Aniston was a very well-known person. Who knows Jennifer Aniston now? All right, at least a few people. Right. And nowadays, you would probably use someone else. But what you can clearly see is that this neuron that they recorded down here in the hippocampus responds here. So each of these dots here is one of these cracking sounds, these action potentials or spikes. And you see a histogram here. So you can clearly see that these images here evoke responses, whereas all these other ones don't. So, and all these images have one thing in common. They are all images or pictures of Jennifer Aniston. This actually not only works for Jennifer Aniston, but it also works for Halle Berry, which I would prefer if it was my choice. But also what's nice with this exact example is that it not only works for images, but even for drawings, even for Halle Berry being masked as Catwoman. By the way, who knows Halle Berry? Okay, and even if you write the name Halle Berry, the neuron still responds. So this is why they coined these so-called concept cells, because they respond to a concept of Halle Berry. They are also concept cells for cars, for oranges, for whatever you might imagine. So this work, by the way, is now continued. One of the postdocs involved in that was Florian Mohlmann, who is now a professor in Bonn. And also one of his former students or postdocs is now going to be a group leader here at the University and Hospital in Aachen, where we will continue this work with them. Now, as I said, I was a young student trying to find my niche in neuroscience. And so I was wondering, after reading all this textbook knowledge, so what is left there? What are the big questions in neuroscience that I could actually focus on? And I found a book called The 23 Problems in Systems Neuroscience, which was published about 20 years ago. And these 23 problems relate to some earlier publication by a German mathematician who 100 years ago wrote the 23 problems in mathematics. So he was German, so it was written in German, but this is still one of the Bibles in math. If you study that, some of the problems have been solved by now. Many are still out there. So I thought, okay, well, maybe that's a good way to start. So I had to look into this book and found this one article in there with this weird title, what is the other 85% of V1 doing? Remember, I told you before there was a Nobel Prize already. I mean, we know everything about the visual cortex. It's in the Kandel textbooks. And so why do they claim that basically we know just something about 10% or 15%? Now, what they base their claim on is several points. One is shown here, that this is how an electrode looks like that is usually used for these recordings that I have shown you. In this case, it's a tetrode because it's full, but they're basically all very similar. It's a wire that you push into the brain, and in the vicinity of 50 to 100 micrometers radius, you can record neurons. Usually you heard this crackling sound, so this was maybe one neuron that was recorded, even though you have hundreds or even thousands of neurons around. So why do you record only this one neuron? It might be the loudest one, or it might be for whatever reason, but we're clearly undersampling here. And this is one claim why we actually might only understand something about the very few loudest neurons. And as you probably know yourself, the loudest ones are not always the smartest ones. So maybe you also want to listen to the other ones somehow. So they formed something like the 7 rules for an ideal experiment. And one was to record from all the neurons without any bias towards the loudest or the biggest, and identify these recorded neurons because they are different ones, as I will show you. And not only the neuron's activity is important, but also the wiring diagram underneath. And you don't want to record only once. Maybe these neurons change completely on Tuesdays or Wednesdays. So if you only record on Mondays, you might just have not the full picture. And what is also important, if you want to understand how we perceive, for example, Jennifer Aniston, it would be important to know if this neuron that you recorded there has anything to do with our perception, or if this is only the product. The fact that I see Jennifer Aniston sparked this neuron, but I actually don't need this neuron to understand the concept itself. So for this, you would have to stimulate or manipulate this neuron, inhibit it or excite it, and see if that changes my perception that I have. So this is called a causal correlation. And you saw in the beginning that the stimuli that have been used were these elongated bars of light flickers and very simple stimuli. People or researchers like to keep it simple because that's what you can explain. However, the brain wasn't made to look at simple stimuli. It's made to look at our natural environment. So this is why you should apply natural stimuli if you really want to understand how the brain works. And finally, usually we don't sit there anesthetized in a head holder. So usually we look around, and most of the images that we see, we might only see once, or at least once in exact that constellation. Even if I look at you now and I turn my head again, that might be a slightly different angle and so on. So it's important to record this activity during a natural behavior task where these neurons usually act in. Okay, let's go through that slowly. So first point, record from all neurons without bias. I already mentioned that there are many more neurons around than what we usually record. So if we think about which space or which volume we want to cover, even if we just take a very small volume of a few hundred micrometers, we already have a few thousand neurons in there. And if you look at the numbers, most of this volume is made up out of neuropil. So these are dendrites and axons. There are about 10% of it, which are cell bodies of neurons and glia cells. And about 4% is blood vessels. So this gives you about 9 times 10 to the power 4. So this is about 10,000 neurons in the cubic millimeter. Sorry, it's 100,000 neurons. And about 10,000 synapses per neuron. So if you put these numbers together, you already realize that this is quite an endeavor to figure out all these neurons, even in a small cubic millimeter as shown here, or even within a few hundred microns. So what we use is a technique called multiphoton imaging, which for sure you have seen yesterday in Simon Musal's talk, I assume. And what that does is that you can actually take a laser scanning microscope to see fluorescence inside an intact brain. So this was in a mouse, scanned from top of the cortex about 200 microns down, and it's about 200 microns side length. So it's much smaller than a cubic millimeter, but already in here, there are a few hundred neurons in there, and the neurons are green. Oops. The neurons are green, and in red, you see astrocytes staged with a different dye. So this was a fluorescent dye put on inside the brain, and then these cells take that up. And in black, these black snaky things that you see there, these are blood vessels. There is nothing in there that is stained. That's why it remains black. Yes?
Sorry, what is the scale on?
So the side length here is 200 micrometers. And so the blood vessels have no neurons inside, of course, so that's why they remain black. What you can clearly see is around the blood vessels, there's this reddish halo. Does anyone know what that red surrounding of the blood vessels is? Any idea? Perfect. Perfect answer to the exam question. Yes. So as I said, red is astrocytes. So it has something to do with the astrocytes. They form this little so-called end feet or protocytes, and they together form the blood-brain barrier. So this is the barrier that makes sure that not every shit that we have in the blood gets into our brain. Yeah.
What's the brand of the brain or the diagonal that's recording, more or less?
So this is a stack which starts at the top and goes down. So,
Okay,
there's no time involved. Yeah.
Okay, that was.
I mean, it took some time to take it, but there is no time information in here. The time information is actually here. So this is same mouse, same staining, but now it turns out that this green dye that I used, OGB, Oregon Green BAPTA-1, BAPTA is a calcium chelator, so it's a calcium-sensitive fluorescent dye that blinks each time there's calcium inside or rising inside the neuron. And calcium rises each time the neuron fires an action potential. So this crackling sound that you heard before, this is now visible as blinking. Same thing. So each of these transients here would be an action potential. And here you have a time bar. You see that this is much slower than what you might have imagined, because action potentials occur on the millisecond time base, but these calcium signals, they are somewhat slower. And in addition, it takes some time to kind of raster this entire image with a laser. So usually we image this between 5 and 30 hertz is our recording frequency here. With electrophysiology, you usually talk about kilohertz. So there is a discrepancy, and this is also why these transients have different heights. So this might have been 1 action potential, and this might have been 10. Nevertheless, we can use this to map neuronal responses, and this is in the visual cortex, and you see down there the stimuli similar to Hubel and Wiesel. I have shown different orientations, and you see that neurons blink specifically for one orientation but not for the other. And what I can do is that I can now map my neurons according to their orientation preference, and so color them here, or the preference for different spatial frequency, different temporal frequency, both together give you the speed. And as you can clearly see, there is tuning for these different properties, similar to what Hubel and Wiesel have already described for cats and primates, with a clear difference that there is no organization. So in a cat or in your brain, all the red neurons would have been in one corner, all the green neurons in another corner, because there's this columnar organization, which is not present in the mouse, because that's what we call a salt and pepper organization. By the way, I think it's recorded, so there might even be a record afterwards. I see. Feel free to take images, pictures if you want to. I also usually put the references underneath if you want to find the original. All right, so this was recording all the neurons. So this we can do with this technique. Now, what about the neuron identity? So neurons come in many different shapes, forms, and functions. So this is just one overview of how the cortex looks like. So this is a pyramidal neuron. These are the main excitatory neurons, and all these colored ones around are the different types of interneurons. Turns out now that even though the excitatory neurons usually look very much alike, they can also be very different if you look at the genetic profile. In addition, these neurons have elaborate dendrites, as shown here. So this is the pyramidal neuron that you see there. Does anyone know what this is? Yes, very good. Perfect. We could continue this now. But so these have very specific dendritic trees. And it's likely that this very specific morphology also drives a very specific function. Everyone recognizes, or nearly everyone, recognizes a Purkinje cell. And so it's likely that this very specific arborization also allows them to do whatever they are supposed to do, even though we don't know that yet. But this is one of the main focuses that I have been working on over the years, also where I started actually my PhD in, and still today I'm trying to figure out what these dendrites are actually doing and how these individual neurons integrate information within these dendritic structures, because this is where all the synapses sit on. So this is again mouse visual cortex. So the surface would be here, and this is about 400 micrometers down into the cortex. And this is the side view of a stack that we have taken, similar to the stack that you saw before. And this is a plane here at the top. And this plane here was taken here and further down. So this is why here you have mainly soma chat. And here you can maybe see the quality is restricted because in order to have some speed for the recording, we have to reduce the resolution. Nevertheless, you can see some dendrites blinking here, and we can analyze this. And this mouse has been awake and has been running on a wheel and got different stimuli. So it's actually a multisensory discrimination task. So there were visual stimuli, tactile stimuli, and the mouse had to tell us on which side are more stimuli. And so we can now follow from the individual neuronal integration to how this is represented in cortex, is learned, and how the decision is made. Okay, so speaking of individual neurons, obviously it's not enough because they are also connected to each other. So it would be nice to also get the neural wiring diagram. And there are several approaches over the last decades where research have tried to get this in its full extent. On the left, you see the Human Connectome project. And this here is from the Genelia research farm, the Mouse Light project. And you can see that there's a lot of wiring. I mean, I told you 80% or even more is wiring. And these neurons actually have very long extensions. So there are neurons that might start here in the visual cortex or even in the cerebellum and project all the way to the front and vice versa. So this is more or less a nightmare to figure out who is connected to who. Nevertheless, there are several approaches to do that on the fine scale level and in both human and the mouse, and also, for example, in Drosophila. So the way we have approached that was a bit simpler. So again, we took our mouse where I did the functional mapping, and then I picked one of these neurons and we approached an electrode or pipette to this neuron. We electroporated it, so we filled it with a dye. And this dye, biocytin, is visible afterwards in the light microscope, so we can reconstruct this neuron. And we can even see it in the electron microscope, which you need in order to find synaptic connections, because synapses are so small that they're just little blobs here. But in the electron microscope, you can find the axon of our stained neuron, and you can see that the postsynaptic side here is a spine. So this was obviously a synapse onto an excitatory neuron. what we found here was actually that most of the synapses close by are on inhibitory neurons. So there seems to be a bias there. Yes.
Does adaptation play a role, an important role in this functional mapping, or is that?
I mean, any kind of adaptation or plasticity plays, of course, a big role because the mapping of the connectivity is usually a snapshot. And it might actually have been very different the next day. And also, any kind of approach that has been done so far to map this over time a bit better shows that there's a drift, and that this drift is still not fully understood how we can actually function, even though our neurons change their connections and their tuning the whole time. Yes. So we can only get rules, but having the full connectome is, I think, actually useless in the mouse. In the fly, it's maybe a bit different, or in the worm, but in the mouse or in the human, I think having rules probably helps more. And actually, since you just raised this point, I think in order to make sense out of this, what we anyway need is to get these rules that we get from our experimental data and put it into network models where we then can play with these diagrams in order to reproduce what kind of result we had in the experiments. And then we can also include adaptation or learning or changes over time. And this is also what we did in a number of studies, and that is our current approach still. And as I mentioned, so our study is a few years older now. Nowadays, there are quite some endeavors to make this work in the mouse to get a full connectome. So the Microns project tried this in a cubic millimeter with 120,000 neurons that they reconstructed on the electron microscope level. And about 1,000 neurons are shown in this image. And about 200 have been recorded. So where they have, with this imaging technique that I've shown you, they have the functional mapping, and now they can find out who is talking to who, and does that have anything to do with your tuning and your responses to visual stimuli. You can imagine that this is a lot of data and involves a lot of people looking at it and analyzing it, and probably a lot of AI to actually handle it and make sense of it. So this is still ongoing project. The main drivers here are the Allen Brain Institute in the US and also the Stanford groups from Andreas Toljes. Now back to our 7 rules. So I have shown you most of it. I have mentioned these ones, so I guess Simon has talked already a bit about these here. So one point that I want to talk now on is the application of natural stimuli. So this was actually the point where I thought, okay, so this hasn't really been done when I started back then. And I found that a reasonable focus to look at. And so I took the mouse and I took a database of so-called natural images. And actually, to cut a long story short, when I submitted my first manuscript to Journal of Neuroscience, I got it back with a reviewer comment saying if Hubel and Wiesel had shown their holiday pictures to the cat, they would have never gotten the Nobel Prize. Needless to say that the paper was rejected. And so I sat down and wondered, I mean, to some extent, I would object. I think they still would have gotten the Nobel Prize, maybe even a better one. But one important point here is if these are your holiday pictures or the ones from Hubel and Wiesel, it's very difficult now to pick which one of these you want to show because they're all different. And how can you quantify that my holiday pictures might for sure be different to yours or to the ones from Hubel and Wiesel? So I actually took one of my holiday pictures and I tried to analyze those to see if I can find some quantification. So this is the Calonge in Marseille. Again, we are in France. And what you can clearly see here is that there are oriented edges, which are in every scenery around you. And there are many vertical and also many horizontal edges. So this is what you see here in this distribution. And there are only very few diagonal or so-called obliques. And turns out that this is the case in all natural scenes and also in man-made environments. If you look around here, there's a lot of vertical and horizontal, not much diagonal. So if I go back to my mouse and I look at the orientation tuning that I have measured in the mouse, I also see that there are many neurons that prefer vertical and horizontal neurons, but only very few prefer obliques. And this is in the primary visual cortex, and even more so in the higher visual area called PM, for posterior medial. Researchers are not very good in coming up with names, so this is why they just take the location. So posterior medial is here next to the visual, primary visual cortex. Now, the fact, so this already suggests that the brain has evolved in order to represent our natural scenery. A lot of vertical and horizontal, so-called cardinal orientations, a lot of neurons that represent them. Makes sense. Also, we tested how good mice actually are in discriminating these orientations. So what you see here is a mouse in a touchscreen chamber. So it has a screen where it has to touch a certain stimulus that is rewarded. And every time it does that properly, it gets a drop of water on the other side. And you can do this with a cardinal orientation. So this versus this, as you see here. But you can also do it with oblique or diagonal orientations. So once we train them, eventually they will do this equally well with both combinations. However, as you see here, they learn much faster for the cardinals than they do for the obliques. So if we train them long enough, the perception will be equally well. But the fact that they have a brain that is tuned for cardinals obviously makes it easier for them to discriminate these two stimuli. Yep.
You're showing the addition of female and male. Have you seen any difference in between the groups?
No. Actually, this is why we have this written there, because one of the reviewers, maybe it was you, asked us this question, and we did not find a difference. But I have to admit, we also were not looking for it, because so we did not have twice as many mice. So we only had whatever we got, male and female. We mixed them usually, and I guess in order to really figure that out, we would have had to have a larger cohort. Here, I think we had 6 animals. So we usually try to stay on the low side because these are animal experiments and the animals have to be water deprived. So, I mean, they're happily running around there. And so I think the impact is not that big, but they are limited in water so that they get their water during the experiment. Otherwise, they would just sit there and not do very much. And there's one difference between males and females. Males are usually thirstier than females are because of their metabolism. And so they are more likely to actually do something in there. It's not that they are learning faster. It's just that they're doing more trials because they have to get this water. And that makes it also difficult then to talk about the differences that you find. Okay, so orientations not only come as the difference in the main orientation, they also come as a distribution. Again, there are the natural scenes. So there is the domination of the cardinals, vertical and horizontal. And but there is a distribution of these orientations. And this distribution is quite broad, as you see here for the entire library, but also for the 2 example regions. So you see that some images can have a very narrow distribution, some can have a broader distribution. So usually you don't have just one orientation that you see. So this is why we were wondering if this distribution also plays a role. So we created stimuli, so-called motion clouds, that have either just a narrow range, so only one orientation, or a very broad range of orientations. And here you see that this is adjusted to what we found in our library of natural images. As it turns out, if I show the stimuli again to the mouse, so you see here from the narrow to the broadband an example, and in color the neurons that responded to these stimuli. You see that there are a lot more neurons responding to the broadband stimulus, and also they have larger responses. And if I quantify this, you can also see here about twice as many neurons respond, and they also have, on average, larger responses compared to the narrow ones. Now, you could argue that, well, this is to be expected because you have way more orientations in there. So you might also just have way more neurons that like these different orientations. So what we then try to figure out is if this additional number of neurons that responds here, if this also helps the animal, the mouse, to perceive these stimuli better. So if this has any impact on the perception of the mouse, if the mouse realizes that there are more neurons that are active. The other option would be that just the neurons realize that there are different stimuli and can discriminate these stimuli better. So what we did is we made a task similar to the touchscreen chamber task that you saw before, where you have to discriminate either different spatial frequencies, where we alternate the bandwidth of the orientations, or we have orientation discrimination, where we alternate the spatial frequency bandwidth. The reason for mapping, for mixing this, is if I do orientation discrimination and I play with the bandwidth, then I would have ambiguous stimuli, because as I increase my bandwidth of orientations, then there's overlap between these two stimuli that have to be discriminated. So this is why we had to separate these features. And if we do that, then you see that the neurons perform better. So the performance of the neurons is measured as the so-called area under the curve for the receiver operator curve. Does anyone know what that is? Now I got you. There's one. So this is the standard measure in signal theory. And usually my exam question is, what does this have to do with Pearl Harbor? So in Pearl Harbor, there were, you remember, Second World War, Japanese were coming, the Americans were sitting in front of their radar machines, and they did not notice because somehow the signal was hidden in the noise. And so this is when a new field started, the signal theory, and where you try to define algorithms how to separate a signal from the noise. And so therefore you have two distributions. So in this case, it would be for the two different responses to the two different stimuli. And then you can measure the overlap and how many false positives and true negatives and so on you get. And from this, you get a measurement, which is called the AUC for the area under the curve. The bigger, the better, basically. And so you see that this is increased. So the neurons actually would have a better ability to discriminate stimuli if they are broadband, so if the distribution is broader. Now we can compare this to the mouse, same touchscreen chamber as before, and also the mouse has at least a slightly but significantly improved performance if we show the broad motion cloud stimuli compared to these simplistic gratings. So to put this together, I have shown you that natural scenes contain broadband orientation and spatial frequencies, and that this increases responses in the visual cortex, which what I haven't shown you is that the explanation for this increase is actually not only that there are more neurons. It turns out that also there is a decreased so-called surround suppression. And I could give you a whole lecture on this. If you're interested, you can talk to me later or just look at the paper. It has something to do also with predictive coding and so on. And in the end, that helps the mouse to actually perceive these stimuli better. So natural stimuli are perceived better than just simple stimuli, presumably because our brain is made for it. Yep.
Question. What about depth? So this three-dimensional component of stimuli.
Yeah. So the mouse has two eyes, as we do, but they are more on the side. So it's more to cover a large visual field. we have the eyes in front, and most of our visual field overlaps. For the mouse, it's only 20%. So depth perception for the mouse plays less of a role, of course. In addition, the mouse has very small eyes, which means that it's difficult to have a very good focal depth. For those of you who play with photography, if you want to do these very nice portrait shots where the background gets blurry, you usually have an objective which is this big. Because the bigger the objective, the bigger the angle that you have, and that reduces your focal depth. And so mice basically have a focal depth between here and over there, and they cannot really focus on anything in between in particular. So that reduces their ability to do what we can. I can focus on you or you in the back. And however, most of the time, we actually can do this with one eye as well as with the two. So this textbook knowledge that we have the two eyes for 3D vision, you probably noticed that even if we close one eye, it still appears very much 3D. And the reason is because our eyes are constantly moving, and our brain uses real intelligence, not AI, but to make a 3D effect out of it, similar to what your smartphone can do these days. You probably have also one of these newer ones. You take a picture, and then you can turn this picture afterwards a little bit to get the 3D effect, even though there was only one camera there. But you can do this disparity tuning, it's called. So you, in the binocular cortex, you have neurons that receive inputs from both eyes, and then you can actually move the stimuli backwards and forwards a little bit and see when is the response of the neuron best. And so there are neurons that have different disparity tuning. And so then through this, you can actually get an idea if something is further in the back or in the front. Okay. So I have talked about a lot now from this book, but one big question that I did not find in the book, and that obviously all of us might be interested in, is how does the human brain work? And one main difference that you see here between the human brain and the rat brain is the size per se, but also that in relation to the size, the cortex in humans is a lot bigger than for the rodents, and also because of the foldings, it has even more surface. And so therefore, you could also just focus on the cortex and to say, how does the human cortex work in relation to the brain, which I would put in as the 24th problem into the book, if I could. So this is actually one focus that has started from in my lab a few years ago, because we now have access to the neurosurgery tissue that I mentioned in the beginning. But this work has already been done by collaborators in Amsterdam. So Natalia Gorinova, who I work with, has published a very nice overview a few years ago. So not only the brain is larger in humans and the cortex is thicker, also the neurons become bigger. So this is a human neuron down here, the cell body, and this is the dendrites, and this is the mouse in comparison. So what you see here is the dendritic recording electrode. So people have looked at dendrites not only by the morphology, but also measuring their function with these electrodes. So the cortex is thicker, so therefore the dendrites have to span a longer distance, but it turns out that these dendrites are also more complex, so they have more branches and so on. And also, if you take these patients where you get this tissue from, you can also do IQ tests with these patients beforehand, which is usually done before and after the surgery to make sure that you didn't cut anything really important. And so this is why you can correlate the different features that you find in their brain with their IQ. And it turns out the strongest correlation is actually with the dendritic size and complexity. So the more complex your dendrites, the smarter you are, or the other way around. I think that already is motivation enough to figure out what these dendrites are actually doing, which is one of the reasons, besides their beauty, why I work on them. So turns out that these longer dendrites also come with a caveat, because you have a much longer cable that you have to cover for the signals to reach the soma. And the soma is the place where everything is integrated. You generate an action potential, and that goes down the axon. And this longer cable also reduces the chance that any signal that comes up in the distal dendrite actually reaches the soma. So in the rodent, there's still some propagation. It turns out that in the human, this propagation is reduced. And in addition, to make it even worse, the ion channel density that you find in human dendrites is also reduced. So these ion channels might actually help to amplify, like, for example, sodium channels or calcium channels might help to amplify this. And so this is also why the input-output function is reduced. So this means for the same input, you get in humans less output than the rodent. So that might all argue that, well, sounds like a disadvantage to me to have these big neurons. So there's obviously something in these neurons that works differently to the rodent, differently to everything we know so far. And this is actually why we work on these neurons and in these dendrites to find out how they actually function, how they integrate. So presumably they do this integration job up here and then only send signals down here that are important and make sure that these signals get through. And just to give you a brief idea how this looks like, so we get the resections from humans, usually from cortex in the temporal lobe, which is down here. So this is below the area that's important for speech and outside the auditory cortex. So this is one of these areas where the memories were placed before. But it turns out if you cut that out, most of the memories are still fully intact. So the impact on the patient is not as huge as if you cut out other parts. This part is usually healthy. So if you have a tumor, it's usually somewhere down inside your brain, and you have to get there to cut it out. So for this, you have to cut a hole into the cortex, which usually can be up to 5 centimeters big. So it's a big hole that you have afterwards. Nevertheless, you're more or less normal, so you survive and you keep your functions. But we have healthy tissue from these patients that we can work on, which is in fact larger than a mouse brain, just as a comparison. So we cut this into slices. You see this here. So this is a block of the human brain about less than half an hour after the surgery. It's on our slicer, and this would be one slice. You can nicely see the folding of the cortex, and you also see the white matter and the gray matter on this image. And then we culture these slices. So we have them in so-called well plates, and we put them in the incubator where we can keep them up to 3 weeks. And the advantage of keeping them for that long is that we have more time to do recordings on them, but we can also interfere with these cultures or with these slices and neurons in there for a longer time. So one thing that we have found already, so this is a study that is in revision at the moment together with many groups and spearheaded by Thijs Verhoek in Cape Town, is that we do a lot of recordings of these neurons. So this is action potentials and stuff that we record from patients that start from infancy all the way up to late adulthood. And what you see here, each dot is a patient or is a recording. Each line here is a patient, and you can see where in cortex these recordings have been made. And what we found is that maturation of the human cortex appears way later than in comparison to the mouse. Of course, the mouse has a shorter lifetime, reaches adulthood much earlier, but basically there's after adolescence or puberty, there's not much happening in the brain anymore. It stays like this. Whereas in humans, most of you are probably nothing like finished yet. And there is, for example, the theory that people enjoy jazz music more in their 30s because their brain hasn't fully developed yet, which is about here. So if you don't like jazz music, don't worry, it will happen eventually. And so as you can see here, there is still an ongoing process even in late adulthood. Yes.
So if someone starts liking jazz in their 20s, means the brain is more developed. But
Yeah, so what people found is that people who like jazz have a certain frontal area that is larger or responds more strongly, and that is usually developing over time. So I would assume that people who like jazz already in their 20s might have already a more developed area there. It's because of this abstract and non-melodic music, which might not sound enjoyable to everyone the same way. Some people will never enjoy it, maybe because they will never reach the, I don't know. And okay, so that means the human cortex obviously develops very differently to that of the mouse. And yep.
The previous slide, what was those metrics mean, more or less?
These here.
Stability.
Yes. So maybe I go back one. So what we usually measure is, so this is in vitro. So we do intracellular recordings with a patch clamp electrode. And what you can do there is that you inject currents. So these are current steps. And then you measure the voltage response. This is the membrane potential that you measure in the neuron. So these are action potentials, as you have seen in textbooks before. So you can measure the action potential amplitude, width, the threshold, the firing frequency, the adaptation. You see it starts fast and gets slower. And also you can use different stimuli like a RAM or a chirp, and then you get all different kind of measures. And so this is a standard range, and this is just a few out of the many measures that have been done. So this is the action potential stability, half width. Tau is the membrane time constant, how fast it charges and decharges. And real base is the current that you need in order to evoke at least one action potential, and so on. So these are, there are more than these, but these are the ones with the strongest impact that have been shown here. Okay. Okay, so I mentioned that we have so-called organotypic cortical slice cultures, so we can keep these slices in a magic solution for up to 3 weeks. Turns out the magic solution is human liquor, so the human cerebrospinal fluid. Unfortunately, not from the same patient. There are patients where this is who get a drainage, so they get some of this liquor removed. The hydroencephalon is one example. So where you otherwise would get too much pressure in your brain because this drainage doesn't work naturally. So there we get quite a lot of this liquor so that we can use it as a serum to bath our slices in. And that allows these slices to feel happier and to stay alive for a bit longer. And because they stay alive for a couple of weeks, we can transfect them with viral vectors containing different genes, for example, for green fluorescent protein. And so we can make these human neurons fluoresce like this and just look at their beauty and analyze the individual dendrites and spines that you see here. So this is done with a deep learning algorithm that we use for that automatically recognizes what is a dendrite and what is a spine. And we can match their shape. And different shapes tell us how big these synapses are, how strong these synapses are. And through this, we also want to map this through a lifetime, but also depending on the lifestyle of the patients before. And obviously also this is related to disease because these spines disappear first when you develop neurodegeneration like Alzheimer's. And as I mentioned before, it's these dendrites that contain somehow our intelligence. So this is where stuff happens when we think. And so this is why we also want to use functional indicators like these calcium indicators that I have shown you before to actually figure out the signals in these dendrites. And the nice thing here is that we can do this with genetic tricks. So we can mark different cells based on their genetic profile. And we can also manipulate them with optogenetic measures. Basically, all the stuff that I have shown you before in the mouse, I can do now in the human, not in the full brain yet, but at least in the brain slices that we have. And all the knowledge that we get from there, we also gather and put them into models. So this is work that has been done by Jutta Perazzi in Crete, where they have built so-called deep learning networks. That's the AI that you have in your smartphone or ChatGPT. And but they have added an extra function, basically an extra layer in there that does stuff similar to what we know about dendrites. And if you put that in and you train this network to discriminate cats from dogs, this is one of the standard tests that you do, you see that the performance with dendrites is much better. But importantly, the energy consumption is a lot less. So it's more efficient because you need a smaller network in order to do the same thing. And I guess you all know that ChatGPT consumes about 20 times more energy than a standard Google search. So if we all use ChatGPT the way we have used Google so far, we would need about 20 times more power on this planet. So it would be important to find something like this here to make these networks more efficient. And this is what happens when you ask AI to what is the effect of dendrites on intelligence. So this is what came out of it. But obviously dendrites can create beautiful images, but also are important for the function of our brain. So with this, I'm at the end. I would like to thank a few people in the lab. So as you have seen, the cherry trees are blossoming at the moment, which is the time in the year for us to make new group pictures. And we call ourselves the Brain Lab Aachen, which is together with collaborators, Toby Bock, who focuses on the hippocampus, Simon Musal, who you might have seen yesterday. He works more on decision-making and brain states, and Hannah Koch, who I work with in the human cortex. And we share basically the same lab and spaces, which is actually very exciting that we can work here so closely together. And that also increases then the size of our group, of course. And all this works because we have some funding that makes this all possible. Thank you very much. And if you have questions, please, I'm happy to hear them. Thank you. Yes.
So first, we will check
Thank you.
I would like to ask you in that you slice your brain, and you make several parts around the measure at the same time, like in several points in the space.
Yes. So a group in Berlin has established that to do up to 8 recordings at the same time because they want to get the wiring diagram. And basically, the idea was to get a patient-specific wiring diagram so that you can do it as often as possible in the slices from this one patient to see if there are patterns that are different between different patients. So if A is connected to B, is C also connected to A and B or not? And you can imagine there is a lot of games that you can play. The connectivity between 2 neurons is roughly 10%. So this is why you need a lot of paired recordings to make any statistics out of it. So this is why they use 8. So we have 4 electrodes, but at the moment we haven't done much of that because it's a very frustrating work, and I'm still looking for a PhD student who has a very high frustration level to do that. Yeah, but a different way to do that would be to record from one neuron and optogenetically stimulate with a laser all the others. So this is the approach that we are taking at the moment. So that exactly is like this. We can get the connectivity with less frustration and just one recording. Then we can't record both ways. We only see the result in one way, but that is one possibility to do this more efficiently. Yes. More questions?
Again, very nice talk.
Thank you.
May I ask you what a specific deep learning algorithm you use for the reconstruction?
This one here. So this particular one is called Respan. So that's a Python toolbox that was published last year that we have used for that. That is based on an NNUNet algorithm, but I have just used it. So that's all I can say about it.
I'm gonna like use a user as well.
Okay.
Can you just repeat the name?
Respan, RESPAN, Respan. We use the black dot here. There are different ones, and this we have tried worked quite well. One other thing that we try to do is also to analyze the shape, because at the moment this is mainly done by humans saying this looks like a mushroom or this looks stubby. And so there are ways to quantify shapes. And there is also a toolbox which does that, which is called Spine Tool. And we are trying to get in touch with these people. Unfortunately, they are in Russia, and officially we are not allowed to work with people from Russia at the moment. And but nevertheless, that seems to be a very promising. So maybe the world will be a better one at some point, and we can actually focus in science. More questions? Yeah, please.
Is there any insights that you could share with us regarding your experience with studying perception that could be used to better influence any research that is going into the direction of studying diseases that alter perception, that have to do with our inability to maybe recognize specific patterns, or when our internal state gets a little bit distorted or confused?
Yeah. I mean, one thing that when I started in visual cortex, I came across is the effect of blind sight. I guess you all might know that there are patients who are blind, but if you put them and they have to walk over there, they will walk around each table and chair without any problem. But if you talk to them afterwards, they have no idea because they didn't see it. And the reason is that the primary visual cortex is maybe after a stroke damaged, but area MT or higher areas still work. So those areas that do the where is stuff processing, so they still work. So the conscious processing is not there, but somehow it's still working. And we all know this also, that if we drive with our car, we might have not recognized the obvious thing that is there because we haven't paid attention. Or also the famous gorilla in among the basketball players. I'm not spoiling too much here, but there is a video that you might want to see. And where some things are there, but we just don't recognize them. And I think, especially if we talk about perception, I think that's very important to keep in mind. And especially with the mouse that we work, we don't know what the mouse actually recognizes or perceives. So this is why it's important to have these tasks where the mouse can actually respond to something, so that we actually get a better idea what the perception of the mouse actually was.
And how long does it take to train the mouse to?
So that depends, of course, on the task. There are tasks that they learn within a day or two. So for the task that we do, so this is how many stimuli are left and right, and there are two spouts, and they have to wait until the spout comes in and not immediately lick, which is something very difficult for a mouse. And so there it takes several weeks, so up to 4 weeks to train them until we can actually do something. It's very different in between mice, even though they are all siblings, brothers, sisters, actually genetically the same. But it's something that always amazes me how different they are in their in their performance, but also in their strategy. Like some just start straight away, but then they decline over time, and some kind of slowly, a bit nervous, and all kind of. It's just like working with a bunch of students, even though they are not all the same, at least genetically. But yeah, so this is something that I haven't figured out why that is so different.
So then the experiments are made when they are kind of adults?
Yeah, so we start usually around 2 months into their age, which is clear adults. Also because we don't want them to grow anymore. So the brain should, because we drill a hole into the brain so that we can look inside. We glue a window on there. And so obviously that should all remain the same size. And also then it's more stable for them to learn as if we start earlier. But if they're too old, it's also a bit difficult. So a 6 months old mouse is a bit harder to train than when they're 2 months old. So if they're in the student phase, they're just okay. Yeah, so a mouse usually gets to 1 year. We have mice that are 2 years old, but in nature, they would never get that old. Yes.
I had a question about the technique, the multi-photon imaging that you showed at the beginning. So is that because I'm not familiar with this type of imaging thing? Is it like, how is it different from light sheet microscopes? And
yes, hold on, I just show
then you
you.
were saying that you move along the axis, so no time involved, right? That particular one.
In that particular, so in order to get this here, I took an image here that looked like this. So I took an image here, then I went 1 step down, another image, and so on. So this might have been 100 images. Each of these images, I took my time, made a very nice one, averaged several times, and so on. And then I took this stack and just looked at it from the side, and this is what I see. These images were done at this plane, but at a higher speed. So this is why the spatial resolution is lower. And with the laser scanning microscope, so that means the laser has to scan across here, back and forth, here probably 500 times to do that. And the computer knows where the laser is. And I detect the fluorescence at this point. So there's no camera, it's only a detector for amount of fluorescence. And then because I know where I was, I can put that together afterwards into an image like this here. And the reason for using this multiphoton trick is because in scattering tissue like the brain with your smartphone, if you look at the top, you can only see the first maybe 50 microns, and then it's just blurry. The laser can actually get up to 1 millimeter into the brain. It's an infrared laser. And the multiphoton effect, that would be another lecture on its own. But the effect basically means that I have a very clear focal depth, and only where the laser is focused at the moment, you can get fluorescence, nothing above, nothing below. So any fluorescence that I measure while the laser is at this point in space can only come from here. And so this trick, there's no blurriness then anymore. And this is why I can actually use this even deep in the brain to get these fluorescence images. Yep. Okay, so as I mentioned, if there are more questions that come up later, feel free to contact me. That's no problem.
Thank you so much, Bjorn, for that.
Thank you.